One of the roles of the liver is to regulate the levels of fats, carbohydrates and protein in the bloodstream. These have reached the liver from the digestive system via the hepatic portal vein. And what the liver does is to process the material that's in the bloodstream to restore levels back to the normal level. So this is an example of homeostasis. And what we're talking about is a process called negative feedback. Negative feedback is a system used in the body and elsewhere, and what it actually does is to ensure that the body system resists changes away from the normal level. And so when we get hot, for instance, what do? Well, we sweat, we become red, and our hairs actually flatten. And all of these things help to actually reduce body temperature. If we get cold, then basically what happens is that our surface blood supply reduces, so we go pale or even blue. We have our hairs standing up, not very noticeable in humans, but it is in animals. And of course, what we also do is we actually thermoregulate. Our body metabolism increases, so we shiver, for instance. And so all of these homeostasis events that we look at are all a case of negative feedback. We'll talk about a different type of feedback in a future unit, but for now we'll stick with negative feedback. And we're particularly going to look at the way that the liver deals with the huge surge in blood glucose that occurs when the blood supply from the digestive tract passes through the liver. So what we do is we draw something called a negative feedback diagram, and we start off by drawing a line across the center of the sheet to represent the normal or desired level, in this case of blood sugar. And the normal levels are measured in units called millimoles, and we're looking at a level between 4 and 8 millimoles per litre of blood. That's the desired range. Obviously, if you've just eaten, it's going to be towards the top end of the range. If you've not eaten for quite a while, it's going to be right down at the bottom part of the range. So let's see what actually happens when this blood supply reaches the liver. So the first thing that happens is that blood glucose levels rise in the blood arriving at the liver. Now this is detected by specialist cells, not in the liver, but in the pancreas. And you might remember the liver, the pancreas and the duodenum are all very close together and closely connected as well. In this case, what happens is that cells in the pancreas, called beta cells, which are in an area of the pancreas called the islets of Langerhans. So the islets of Langerhans are little clusters of cells, and one of the types of cell found there are the pancreatic beta cells. And these release a hormone insulin when blood glucose levels rise. Now, most people are probably aware of this, and generally speaking, people would assume that what insulin does is it breaks down the blood sugar and brings it back to a normal rate. But a moment's thought shows you this would be incredibly wasteful. We've just spent a lot of energy and a lot of time digesting food precisely to get glucose. What we need to do is to make use of it in a way that's not going to harm us. And so what insulin does is it increases the uptake of blood glucose from the bloodstream. It actually converts the glucose into a more inert molecule called glycogen. And if the glycogen stores of the liver and muscle cells become full, then any balance of blood glucose that needs to be stored is stored as fat. So these are shown on the screen under effects of insulin. The overall effect of this, in normal cases in healthy individuals, is that the glucose levels in the blood leaving the liver via the hepatic vein are actually back down to normal. So they're higher than normal entering the liver, lower and back to normal as they leave the liver. And so you can see over on the right-hand side of the diagram, there you are, the green arrow shows that levels have returned back to normal. What happens if we've been fasting or dieting and blood glucose levels fall? Well, they can do that. A typical case would be that great long gap between our evening meal and breakfast. And if we miss breakfast, then we get towards lunchtime and we're beginning to feel distinctly cheesy, distinctly weak, possibly a little bit sweaty. And what's happening is we're exhibiting a mild form of hypoglycemia. So let's have a look and see what happens. So on the diagram you can see there's a green arrow showing the fall in blood glucose levels. And this time another group of cells in the islets of Langerhans of the pancreas are activated, and they release the hormone glucagon in increased quantities. What does glucagon do? Well, broadly speaking, the opposite to insulin. And so basically the glycogen that is stored in the liver cells, but not the muscle cells, is converted back to glucose and released back into the bloodstream to boost those blood glucose levels. If there's not enough glycogen to actually restore levels to normal, then the body very cleverly will actually convert stored fats and proteins into glucose. Remember that all of these molecules contain carbon, hydrogen and oxygen, so the body can actually do a conversion job quite readily. And so that helps to boost blood glucose levels back up to normal. You can see the red arrow going up at the right-hand side of the screen. Now it's important just here to note that we've talked about insulin and glucagon being produced by the pancreas. These are released into the bloodstream, and for that reason they're known as endocrine hormones. However, we know from our study of digestion that the pancreas also releases alkaline juices and enzymes into the pancreatic duct, which then goes into the duodenum. And in that case, the pancreas is acting as an exocrine gland. So endocrine glands release their products into the bloodstream. Exocrine release their products into a cavity or into a duct. You might be able to think of some other examples of exocrine glands. Now, there are just some terms which it's useful to know. When insulin activates liver and muscle cells to convert glucose to glycogen, the proper name of that process is glycogenesis. So think of Genesis, first book of the Bible, all to do with creation. Glycogen, you're making glycogen. The second word that you need to know about is when the opposite happens and when glycogen is broken down to glucose. And there's a word in biology called lysis, which means breaking down. And in this case, the glycogen is being broken down into glucose, which is then released into the bloodstream. Glycogenolysis. So glycogen lysis. And the final word that you need to know about is when the stores of fats and proteins are mobilized, and that is the making of new glucose. So gluco for glucose, neo means new, genesis means making, creating. Gluconeogenesis. Worth knowing those terms and understanding them, and you'll find them in the glossary of the e-book if you've got that. Finally, don't think of this as an either-or situation. The body is very good at doing these balancing acts, and what's actually happening is that glucagon and insulin are continually being released from the pancreas. But it's the relative amounts and proportions that change. So it's like having a mixer tap. With a mixer tap, you can turn the hot bit up, or you can turn the cold bit up, or you can turn either of them down. And the output, which in the case of the pancreas goes into the bloodstream, the output actually then changes. So it is a very delicate process. I hope you found that interesting.